So thank you very much for this opportunity, and I'm very happy to share the recent research topics. So today, let me talk about our findings of the giant extrachromosomal elements from the human oral microbiome. So as you know, the human microbiome composed of the various genetic elements. Of course, the bacterial chromosome is the major components of the human microbiome, but you know there is extra chromosomal elements such as the plasmids and the bacteriophages, and the types of the elements significantly change the composition of the bacterial communities and influence the bacterial functions. So the several recent efforts successfully identify a large number of novel plasmids and the bacteriophage genomes from the huge data set of the human metagenomic sequencing dataset. But most of the computational algorithms to identify such kind of the genetic elements are trained by the very limited reference genomes. So based on these algorithms, we think that we probably overlooked the element that was highly novel and not even partially included in the reference genomes. So in this study, we aim to discover the previously unrecognized extrachromosomal elements without the reference genomes from human microbiome. So to this end, we apply the long-read metagenomics strategy for the oral microbiome. So in this study, we collected the 56 saliva samples from 3 Asian countries, and we performed the long-read sequencing using the nanoparpomethylation sequencer. And we performed the de novo long-read assembly using the Metafly and obtained a very large and continuous contact dataset of the oral microbiome. So to find the unrecognized novel genetic elements, first we performed 2 classification of all contexts to the known genetic elements. And most of the contexts were classified into the bacterial chromosomes, phages, and the plasmids. But we found that the 6,800 contexts were unclassified into the known genetic elements. So we particularly focused on this fraction of the contexts. And interestingly, within this unclassified context, the 85 contexts have the more than 50% genes that are unaligned to the large protein database. So we think that these contexts are the strong candidates of the previously unrecognized genetic elements, having a lot of the functionally unknown genes. So we focus on these contexts, and we clustered based on the sequence similarity of these 85 contexts, and we obtained 19 context clusters, and we compared the abundance and the prevalence and the cluster size of these 90 contexts, and we found that one of the context cluster, which is a cluster number 1, is the highest abundance, prevalence, and the cluster size. So we particularly focus on this cluster number 1 and try to figure out the characteristics of the context in this cluster number 1. So, we found the technically important things to identify this cluster. That is the fragmentation in the short-read assembly. So this is the one example of the context in the cluster number 1. And this is 300, the total ranges of the context. And the short-read assembly were fragmented in 5 positions. And interestingly, some of these fragmented positions were overlapped with the insertion sequences encoded in the large context. So this is a very small mobile genetic element of the microbial genomes. And another fragmented positions were overlapped with hypervariable regions in the genomes. So most of the context in the cluster number 1 is highly fragmented in the short read assembly due to this type of the complexity of the genomes. So this is the technical advance of the long-read metagenomics for identifying the novel genetic elements from the microbiome dataset. So based on the characteristics of these novel genetic elements, we named this element Inocle. And the Inocles have common genetic signatures. One of that is that they have insertion sequences and found from oral microbiome. And all of the Inocles are circular genomic structures. So totally, we found 29 closed circular Inocle genomes. And based on the GC-skew analysis, we found the replication origin and the terminus from the all Inocle genomes. And another the particular signature of the Inocles is their giant genome size, and we found that the maximum genome size is 395 kb of the Inocle. So next, to identify the host bacteria of Inocles, we aligned the genes of Inocle to the bacterial chromosomal database, and we found that the average 50 ORFs aligned to the Streptococcus chromosomal genes. So based on these data results, we now speculate that the host bacteria of Inocles are Streptococcus. So then we compare the nucleotide composition between the Inocles and the Streptococcus chromosome, phages, and the plasmids based on the tetranucleotide frequency. And we found that basically the Inocles have distinct nucleotide composition compared with another genetic elements. But also we found that the Inocles are more similar to the Streptococcus plasmid compared with the phages and the chromosomes of Streptococcus. So based on these results, we now have a hypothesis that the Inocles is a novel giant plasmid-like elements of the Streptococcus. So also we identified the marker genes of the Inocles, and we identified the 4 taxonomies of Inocles based on the phylogenetic analysis of the marker gene of Inocles. And we named these 4 Inocle taxonomies as Inocle alpha, beta, gamma, and delta. And we also analyzed a large data set of the salivary metagenomic sequences all around the world and identified that Inocle alpha is the dominant and the prevalent taxonomies among the all Inocle taxonomy. And the average prevalence of the Inocle alpha is 52% in the world. So the genetic element is highly spreading and prevalent, the novel genetic element in the oral microbiome worldwide. So the next question of the Inocles is what is the function of the Inocle? So we conducted the functional annotation of Inocle genes. But I mentioned that 95% of the Inocle genes did not have the non-functional genes. So it's very tough to analyze the Inocle functional genes, but we could predict the subcellular localization of the Inocle genes. And we found that 40% of the genes of Inocles were cytoplasmic functions. And within these genes, we found several non-functional genes. And interestingly, we found that most of that genes are related to the stress resistance genes, such as DNA damage repair or stress response regulator and oxidative stress suppressions. So, and also we found that 20% of the Inocle genes were related to the cell wall or membrane associated genes. And within these cell wall related genes, we found the peptide glycan biosynthetic genes from all Inocle genomes. And we also found a series of proteins which contribute to the anchoring of cell surface protein to the peptide glycan. So Inocles possibly potentially have a cell wall biosynthesis and modification function. So finally, we analyze the association between the Inocles and the human deep phenotypes. And to this end, we perform the plasma proteome analysis between the Inocle alpha positive subject and the negative subject. And we found a lot of the pathways were enriched in the Inocle alpha positive subjects. And particularly, we found that the most enriched pathways are related to the immune responses to pathogenic bacteria and virus infection. And also, very interestingly, we also found several pathways which were enriched in the Inocle positive subject were related to the cancer-related pathways. So based on this result, we proceed the association analysis between the Inocles and the cancer patients. And we found that in case of the head and the neck cancer patient and the colorectal cancer patient, the prevalence of Inocles in the aura was significantly decreased compared with the healthy controls. But in case of the pancreatic cancer and the rheumatoid arthritis, that trend is not observed. So this is a summary of the Inocles. So we identified novel giant plasmid-like elements of the Streptococcus, and potentially the Inocles expand the adaptive capacity of the multiple stresses, such as the cytoplasmic stress and the extracellular stress, including the inflammatory responses of the human cells. So this work was mainly performed at Professor Yutaka Suzuki's laboratory at the University of Tokyo, and also we collaborate with a lot of the collaborators. So thank you for kind attention.